Welcome, and thank you for joining me for today's talk on achieving reproducible mouse studies. My name is Dr. Caitlin Gillen, and I'm a technical information scientist with the Jackson Laboratory. Today I'm going to talk to you about how to design and successfully carry out robust mouse studies, as well as how to communicate methods completely and accurately. But before we get started on that, I'd like to talk a little bit more about the Jackson Laboratory. So, the mission of the Jackson Laboratory is to discover precise genomic solutions for disease and to empower the global biomedical community in our shared quest to improve human health. And we really do that through three major ways. First, we have our own research staff with their own private funding that investigates the genetics and biology of human disease using the mouse model. We also provide scientific services and resources to investigators around the world. And we also are very committed to education outreach. And so, in addition to the talk that you're attending today, we also have traditional conferences and hands-on workshops, as well as other webinars and Mice Tech Talk that you can watch online. We also are the world's largest repository for mouse models of disease. We have several locations in the United States, and we also just recently opened a location in China. So you can see our different locations. They're the little white dots there on the map. And so in our time together today, I have a couple of goals for this talk. The first is that I want you to be able to think about and understand what your specific goals are as a researcher so that you can carry out the most successful research in your studies as possible. And so in order to do this, I want to provide you some information to be able to select the most appropriate mouse models and controls for your research, as well as thinking about the different assays that might be the most meaningful for answering your research questions. And then I also want to talk about the importance of communicating methods. So as you can see in this cartoon, and as a researcher, I'm sure you've probably experienced this at a time, when you came across a research publication that you thought was really great and was going to really help push your research further. But then when you looked at the methods section, you realized that some really important details were left out and you really weren't able to replicate what the original researchers did. I also want to hit on relevance for this reproducibility crisis. So a recent study looked at the $114 billion that's spent annually on life science research and development. They saw that about half that, or 56 billion, was spent on preclinical research. And then half of that 56 billion, or 28.2 billion, was spent on studies that were irreproducible. So they were missing information in their lab protocols, their data analysis and reporting, study design, or information about the biological reagents they used. And so because these studies are irreproducible, this can lead to high attrition rate of novel therapeutics. So meaning we're not seeing therapies come out of research. We also can save time and a lot of money if we improve our reporting methods, as well as we can improve our ethical animal use. And speaking of ethics, I would also like to briefly touch on the 3 R's in experimental design. Those are replacement, reduction, and refinement. So replacement includes methods that avoid or replace the use of animals. And so this could include maybe using human volunteers. This could be tissues or cells. You could also consider using mathematical and computer models. Reduction includes methods which minimize the number of animals per experiment. So I'm not going to spend a lot of time talking about how to calculate sample size or some of the statistics involved with that today. If you are interested in that, though, definitely reach out to us and we can get you more of that information. I'm really going to spend a lot of time focusing, though, on proper strain selection and complete reporting of methods. And finally, the third R is refinement. This includes methods that minimize the suffering and improve the welfare of animals. And so today I'm going to focus on specifically avoiding stress, so in handling and housing and in health practices. Now, you might be thinking about the fourth R, or the other R, known as reproducibility. And so there's two parts to this. First, replicable. So this is going to be the precision within an experiment, and this relies on nothing changing. So the methods are going to be the same every time. The reagents that you use are going to be the same every time. Reproducible, however, means that these experiments are going to be able to be reproduced across labs and across publications. And this really relies on parameters that can change, yet still support really one robust biological truth. And so the first subtopic here that I want to really dive into is selecting an appropriate mouse model and control mice for your research studies. So one of the most common questions that we get at JAX is, what is the best mouse model to use for your studies? And so I'm going to give you some things to think about when deciding that. So, like I said, one of the first decisions that you're going to make when designing an experiment is choosing an appropriate model. And how you do that is going to depend on several factors. And the first thing is using science, the primary literature. You want to look through what's already been published, and you want to see what models have a lot of support. So that meaning there are a lot of references for that model. There might be some baseline information already in the literature that you can use. The strain that you might be looking at could already be characterized. And so you're probably really familiar with PubMed already. I'm sure most of you use that regularly. But I want to point out two additional resources that have been really helpful. One is the Mouse Genome Informatics database. This is where you can look up a particular mutation, see if there are any strains available. References will be linked to different strains and mutations, and it's just a really good place to start some of your searching. Another database that's really great is the Mouse Phenome database. And this is where you can look up some basic baseline data for different strains, and you can just find out a lot of really great additional information there as well. The next place you might consider looking after you have decided on a mouse model would be the different mouse repositories that are out there. So how are you going to get your mice? Where are you going to find those mice? And so here I've listed some of the more popular mouse repositories. Of course, the Jackson Laboratory and the MMRRC, as well as some of these other mouse repositories. Now, maybe you've searched through all these mouse repositories and you can't find the mouse model that you want. Maybe there is a particular mutation, a knockout that you're looking for, and it just doesn't exist. So at the Jackson Laboratory, we can actually help you to create that model. So you can discuss with some of our scientists on the model that you'd like to generate, and we can actually help you do that. Now, one thing I want to talk about is disease mode of action. And it's really important to consider that the relevance of the model to the human disease. So the model needs to mimic the human disease symptoms, but it might not follow the same mode of action. And so a single model might only be able to partially reconstruct a human disease. And so one example of this is for human type 2 diabetes. And so we have the DB-DB, the 642, and the OB-OB, the 632 mouse models. However, there are very few people who actually have a single mutation for diabetes. And so for this reason, it may not make sense to use this model to understand how diabetes works in humans, but it could make sense to determine if there's an experimental drug that could manage the symptoms of type 2 diabetes. So really think about considering multiple disease models and determining whether they are going to be relevant to your hypothesis or the therapy that you're interested in. So let me give you another example of this. It's really important to know what your mouse model can do and what it can't do. So this example here is our Nod-Schilt-J type 1 diabetes mouse model. And I want you to note the onset of the disease depends upon the age and sex of the strain. So you can notice here that females have an earlier onset and a higher incidence rate than what the males do. It's also important to note that not all of these mice are becoming diabetic. So there's sort of this range. Some mice are going to become diabetic, some mice won't. And that's sort of what happens in the human population as well. Now, I also want to talk about disease prevalence and severity based on sex. And so the disease prevalence and severity may differ. And so two examples of this would be the lupus model that we have. Lupus is typical to female patients. However, our mouse model uses male mice. We also have our Rett syndrome model, and these patients are primarily female, but some MECP2 mutants have different phenotypes depending on whether or not you're looking at males versus females. And there might also be a sex skew within the published literature. This could be for historical reasons or logistical reasons, something other than biological reasons. So for example, within neuroscience, male animals are used 5.5 times more frequently. However, there's really no more variability within females than males. So it might be that it's easier to do, let's say, a surgery in male mice because the mice are bigger. Or maybe in female mice, because they weigh less, you could use less drug. Are just two examples of that. So model selection, again, with regards to sex, you should really consider using both sexes. And in fact, now NIH grants require you to come up with strong justification if you're only going to use one sex. So I highly recommend that you really think about using both sexes, because you might actually lead to a better understanding of sex differences if that's applicable. You may not realize that there is going to be a sex difference. Both sexes might be needed to more accurately model human disease. There are many human diseases that affect both sexes. And by using both sexes, you can really reduce breeding cost and animal use numbers. So for example, every time that you use 50 male mice, 50 female mice were also bred in order to generate those mice. And so just remember that genetic model does not always equate to disease uniformity. So think about this as more of a normal distribution of a phenotype of a disease, and that age, sex, genetic background, as well as environmental factors can impact the phenotype or the disease that you're interested in. And so you may need to adjust your experimental design in order to accommodate these known characteristics. So you may need to consider increasing your sample size, maybe using more than one model, just to give you a couple of examples. And so next I want to move on and discuss about control selection. And there are really lots of ways to pick controls, some of which I'm sure you're already very familiar with. But today I want to focus on selecting genetic controls based off of genetic background. And so here you can see on this slide that there are two images of C57 black 6 mice, the B6J and B6N substrains. And you might be asking, why does that really matter? And it's because that these are two different substrains with two different phenotypes. So let me give you an example of why knowing the genetic background and substrain is really important. So genetic background is known to influence phenotype. And in this example, which comes from the mouse phenome database that I mentioned earlier, looks at the FVB strain. And this was a model for mammary tumors. And what they did was they took transgenic males and bred them to different female inbred backgrounds. And then they measured the tumor growth in the offspring. And what they found here is that depending on the genetic background of the mother, would contribute to the phenotype of the offspring. And so you can see here in the first column, if the female was a MOLF EIJ bred to a male FVB transgenic mouse, the offspring took much longer to develop tumors than the control FVB mice highlighted in green. Or on the opposite end of the spectrum here, mice that were back crossed one generation to the ILNJ female showed a much more rapid increase in tumor development. So all of this can really drastically change how you might consider designing your experiment, because in one case, it's going to take about 85 days for tumors to develop, and in the other case, around 40 days to develop. And so this is really a much more extreme example of what can happen, but it's really important to consider when designing your experiments. So there are more subtle differences that come into play when we consider genetic background and genetic drift. And so the potential for genetic drift to differentially affect two populations of a given strain maintained in isolation from one another is why we apply the substrain designations to them. And so by definition, a substrain is a branch of an inbred strain that is known or suspected to be genetically different from the parent colony. And substrain designations are applied to colonies when they have been separated from the parent colony for about 20 generations or more, or when there are known phenotypic differences between the two populations. And so when you follow nomenclature guidelines about substrains, these substrains are designated by adding a / after the strain name, followed by the lab code for the investigator or institution where those mice are being maintained. So you can see here in this example, the parent strain is CBA. The first substrain listed is CA for the Carter lab, and then was transferred to GN, or the Green lab, and then to the Lane lab, or LE, and is now maintained by the Jackson laboratory. So you might be asking, how do we lower the impact of genetic drift, and how do we maintain our phenotype to the best of our ability? And one of the most impactful things to really do is to know the history of your mouse. And what I mean by that is to know the genetic background and know the substrain of your mice. So you might consider purchasing cohorts from mouse repositories because they maintain genetic quality. It can be very challenging to maintain similar genetics when you're breeding yourself. Also, if you obtain a strain from a collaborator, you should really get as much information as possible from them to verify the genetic background, and verify the genetic background if possible. And so you can use tools like genome scanning or Gigamuga um, to try to verify that background if you're unsure. And so sometimes it matters whether or not you are using male versus female for the trans gene. And so you want to also keep that in mind as well. And so in summary, I want to just touch on a few things that will help with your colony management practices in order to minimize the impact on phenotype. And so you want to avoid accidentally selecting for mild or strong phenotypes. And you can do that by maintaining defined breeding rotations and mating schemes, keeping a pedigree book, or I know a lot of people use Excel spreadsheets now. You want to refresh your genetic background regularly. Also consider cryopreserving unique strains. So if genetic drift does occur, you can pull stock out of the freezer. You want to try to avoid copy number variations with strict genotyping practices. And as always, consider purchasing cohorts for your experiments from a trusted vendor that minimizes genetic drift. And so I want to come back to this slide and move on and talk about choosing quantifiable assays and meaningful readouts. And so it's really important to consider which assays are going to be better for testing certain models and think about which ones are going to be better than others. So for example, if a strain is known to go deaf over time, you wouldn't want to use them to assay startle responses because they're not going to be able to hear those tones. You want to think about using repeated measures on test subjects because they might lose sensitivity or they might gain experience from repeated experience to the assay. So for example, some mouse strains can actually learn how to use the rotorod. So you might not be able to tell if you're using an automated assay if those mice are falling off of the rotorod or if they're learning to jump off of the rotorod. If you're using an automated method, that method may not be able to tell the difference. If you are manually scoring that, are you sure that they jumped off or that they fell off? So you might want to consider using complementary assays or adjusting your experimental design to account for some of those things. And the assay that you choose might need to depend on the relevance to the human population. So for example, metastasis assays don't really test metastasis, but rather dissemination. And therefore, the assay doesn't really fully model the disease process. And so these metastasis assays have limitations in that the metastasis results from a tail vein injection or a subcutaneous transplant. And that doesn't really mimic the human response. Additionally, the mouse assay might not match up exactly to what's being measured in human patients. And so it might be necessary to develop a new assay in order to better recapitulate the human measures. And you can also work to improve your handling practices to decrease stress. But if there's experimental reasons for certain practices, be sure to keep good notes in order to help you later. Um, Try to keep the experimenters consistent and the handling to a minimum if at all possible. Changing experimenters can be stressful for the animals. And this may mean splitting up groups, performing sensitive assays early in the day, things like that. And so finally, in our last few minutes here, I want to quickly discuss best practices for communicating methods. Um, so here in this example, there was a survey of 271 publications, um, with 72 of these studies conducted in mice. And there were key methods and results, or I'm sorry, there were omissions in key methods and results with regards to strain, sex, age, whether or not the study was randomized, and what the statistical methods may or may not have been. And so there's nothing more important than accurately and completely reporting your research. And so in order for your work to be reproducible, researchers need details in methods and results and the statistical analyses that are performed. And so the NIH guides us to exemplify and to promote the highest level of scientific integrity, public accountability, and social responsibility in the conduct of science. And so we suggest using the Animal Research Reporting of In Vivo Experiments, or the ARRIVE guidelines, as a tool to improve your communication and reproducibility. And so these guidelines were designed to help researchers report their research to the scientific community and to increase scientific rigor and transparency in scientific details. And so these guidelines provide details on what you should report in each section. This is just a brief description of what goes on in each section, and you can see a full description at the link that I provided on the slide. And many journals are also endorsing the use of the ARRIVE guidelines, specifically in this example, Nature. So I believe that that's going to be coming more popular here in the future. And so the final thing I'd like to leave you with is what happens if you still can't reproduce results? And there's several things that I'd like for you to consider. I would highlight here in this example, this is a figure from a publication that looked at experimental results in their lab in Seattle, and then once they moved to their new lab in Cincinnati. The only thing that really changed between these two experiments or groups of experiments was the location. The researchers conducting the experiments were the same, the reagents were the same, and so geographic location likely played a role here. Also consider the possibility that the original findings are just not reproducible. And then once you've exhausted all of the possibilities, consider publishing negative or alternate findings. And so to summarize, reproducibility really starts with a thorough understanding of your mouse models, and that you might also be required to characterize your mouse model prior to conducting your experiments. You might need to take into account for many factors that can influence the consistency of your experiments. And consider following the ARRIVE guidelines so that your research is well reported and can contribute to building a strong foundation for future research. So with that, I would like to thank you. Please feel free to reach out to micetech@jax.org with any future questions, and we look forward to hearing from you. Thanks.